Uterine leiomyomas, or uterine fibroids, are growths that are benign or non-cancerous. They can grow inside, outside, or within the walls of the uterus or uterine cavity. Women with these tumors can experience painful symptoms, but many do not experience any at all. Some of the symptoms are enlargement of lower abdomen, heavy bleeding during periods, which may also cause anemia, frequent urination, lower back pain, and possible reproductive issues. Fibroids are the cause of about 200,000 hysterectomies a year, which is the surgical process of removing the uterus. There isn't an exact known cause of fibroids that can be specifically targeted to treat or eliminate, which is what makes them interesting to research on. Regardless, there are known risk factors according to the research article, A Trans-Ethnic Genome-Wide Association Study of Uterine Fibroids. Having African or African American ancestry, a high body mass index, and aging are factors of fibroids, according to the article. The research study was focused on finding the similarities with genetic variants associated with uterine fibroids between African and European ancestry groups. SNP. With no prior knowledge, fibroids have been said to be an issue for African American women, but this was determined by GWASPs being conducted with small numbers of women of African ancestry. In the research article, there was evidence gathered by a two-stage trans-ethnic genome-wide association study data, or GWASP, and natural language processing to support the genetic variants that were associated within these ethnic groups. These were different groups. The different groups that were tested were from the UK Biobank in the UK and Electronic Medical Records, EMR, in the United States. Individuals that were diagnosed with fibroids, those who didn't have fibroids but had family or ancestral history of fibroids, and those who had gone through 2 or more fibroid screenings resulting in no fibroids finding and with no family history of fibroids were the control groups. SNP, or single nucleotide polymorphism, were aligned within each group I previously stated, and within those groups they tested each race, European and African, and genotyping platform-specific GWASP in 37 reference populations of the respective allele frequencies. Associations between genotypes and the presence of fibroids were found by using multiple logistic regression. GWASP data was concluded with the GPGE weights of genes within skeletal muscles and uterine tissue. With conducting several tests, there were positive and inverse relationships between gene expression in uterine fibroids that were also linked with African and European ancestry. RS47853884, the SNP that was analyzed, had been found to be associated with 12 out of the 13 assessments of the study dealing with race and site specifics. Some of the findings were the increased HEATR3 gene in the skeletal muscle and uterine tissue was found to be positively associated with fibroids across both ethnicities. A variant in the WNT4 CDC42 had different effects in African and European women that were associated with fibroids. Also, TP53 variants were found to be polymorphic in European women and not in African women. These discoveries are consistent with what I stated previously. Having African ancestry would increase your chances of suffering from fibroids compared to their counterparts, including people with European ancestry. But this study shows us that there are shared genetic markers amongst African and European women that are associated with uterine fibroids.